The next presenter is the amazing Thanasis, a jack of all trades. He can perform very well under stress conditions. So that's why we gave him the most difficult project. His title is Genome-wide Association Studies, GWAS, in an effort to provide insights into the complex interplay of NRs, nuclear receptors, transcriptional networks, and their contribution to maintenance of homeostasis. So pretty much it covers everything. So please, Thanasi, enlighten us.
So can you hear me? It's
Yes.
all good?
Yes.
And I have shared my screen, so it's all visible, as far as I know. So let's start. Good day, everyone. Our study is a genome-wide association study, which fixes on nuclear receptors transcriptional networks in regard to the maintenance of homeostasis. So let's begin. What are nuclear receptors? Nuclear receptors are ligand-dependent transcription factors, i.e., they're proteins that partake in the regulation of gene expression. And they have a large number of, they partake in a large number of biological processes. Some of those processes are the immune system, our development, and our metabolism. So we take as prime example of a nuclear receptor, we take the glucocorticoid receptor, which has been studied a lot. And let's bring the example back to what we said before. It does partake in the immune system. It has pro-inflammatory and anti-inflammatory behavior in the system. It does affect development, especially since glucocorticoids are responsible at the time of birth, are responsible for later on the plasticity of the brain to various situations. And of course, metabolism, we all know that glucocorticoids can affect metabolism since they can stay in lipid cells and affect the development of different diseases. So what is homeostasis? Now, let's think. An organism, a living organism, has to cope with various signals. Differences in temperature, from stressful events like loss of a job, to more common things, let's say, like a disease or something. So every living organism has developed a mechanism that retains an inside equilibrium. Either that is physiological, either it is chemical, either it is psychological. And that equilibrium is called homeostasis. So every organism tries to maintain the equilibrium through various mechanisms, since apostasis, which is called when homeostasis cannot be sustained, can lead to various pathological conditions, from psychological problems, let's say a lot of stressful events can lead to depression, to problems with the immune system, like let's say an inflammation that goes on for too long can create different problems later on. So what we did is that we thought, since the glucocorticoid receptor, which has a great part in homeostasis, and nuclear receptors as a whole partake in the regulation of homeostasis, it would be a good place to research and find new information. Now let's go. Let's think that in 2001, when the Human Genome Project was finished, the cost per genome for sequencing was $100 million. So after all these years, it is really impressive, since based on technological theories, we expect each year to double the efficiency and halve the price. And we see this line. But the price has dropped remarkably, which allows us to make more in-depth research. And all this sequencing that can be done in nuclear receptors, as we're trying to do in this project, leads to big data. Now, it's great, we have big data, that helps us a lot in science. But the thing is that we have to extract the correct information, because a lot of this is useless. There are specific things we have to look into it so we can get results. And this is where genome-wide association studies come into. These are studies that study exactly a specific trait or a disease or a specific gene and the way it associates with various single nucleotide polymorphisms. So let's say that we have tall people and short people, specific nucleotide polymorphisms that are absent in some of them and that are present, let's say, in all tall people, so it partakes in that mechanism. So when it comes to homeostasis, we can search each and every nuclear receptor, and let's take glucocorticoid receptor as an example, in conjunction with other cofactors and see if differences, if specific SNPs influence other cofactors to present other SNPs or the function of other cofactors. So let's think it as a key lock system, that we find the key and unlocked, let's say, the secrets of the function of nuclear receptors. So our current study has fully sequenced some Japanese individuals, that's where the flag comes from, in which we have sequenced nuclear receptors, factors that partake in histone modifications, specifically histone deacetylase is a great example. We have all nucleus receptors, as said before, and proteins related to homeostasis. Here we have proteins that partake in the immune system, and like the HLA complex, and proteins that are more of metabolism-wise, partaking metabolism-wise mechanism. So what are the goals of this project? Like we said before, we want to find associations between the nuclear receptors, the cofactors, factors that partake in epigenetic mechanisms, and factors that partake in metabolism. So we want to find, as the jigsaw piece shows there, want to find patterns. We want to find things that can bring us new evolutionary information as how the mechanism of homeostasis came to be. And we want to find some specific associations that may later be used in therapeutic models or have other clinical and laboratory applications. So what are these applications that we may be talking about? Prevention. Let's say we fully sequence your genome. We can then see specific SNPs in your genome that affect the way your organism can cope with stressful events. So we know that you are predisposed to general anxiety disorder, and we can tell you that you have to avoid specific situation. Another one is that you may have some SNP on the glucocorticoid receptor that affects your metabolism. So we can say that specific food is not recommendable for you. Another one is diagnosis. Now there's a specific syndrome, the Chrousos syndrome, with Mr. Chrousos being part of this project actually, which has a pseudo-Cushing phenotype. And these people have problems in their metabolism function. So when we see something like that, we may think that this is a Cushing syndrome. And we may say that, but that diagnosis is wrong. So we can fully sequence, as said before, by using your genome, we can make more exact diagnosis and thus save time because a wrong diagnosis consumes a lot of time that could be used for therapy. And since we said therapy, let's go to the final part, which is personalized medicine. We know the SNPs. We know that these SNPs influence homeostasis. So we know that this person can take specific drugs that influence his homeostatic mechanisms and help him. Let's say somebody has a SNP in the glucocorticoid receptor, and drugs that target the glucocorticoid receptor cannot affect that specific person because of that SNP. So if we know that that person has a SNP, we can search for a different drug, one that is more potent and has lower toxicity. So I made this a bit quick, but I'm expecting a little less in time actually. But if you have any questions at all, I'll be glad to answer to them.
Perfect. Thank you very much, Thanasis.